Hello, myself Chaitra M Hebbal from Gadag, presenting the PPT on topic biofertilizer. Biofertilizer definition. Biofertilizers are defined as preparations containing living cells or latent cells of efficient strains of microorganisms that help crop plants uptake of nutrients by their interactions in the rhizosphere when applied through seeds or soil. They accelerate certain microbial processes in the soil, which augment the extent of availability of nutrients in a form easily assimilated by the plants. Use of biofertilizers is one of the important components of integrated nutrient management, as they are cost effective and renewable sources of plant nutrients. To supplement the chemical fertilizer for sustainable agriculture, several microorganisms and their associations with crop plants are being exploited in the production of biofertilizers. They can be grouped in different ways based on their nature and functions. Nitrogen fixers. Free living nitrogen fixers are aerobic Azotobacter, Bejernic, and Anabena. Anaerobic free living nitrogen fixers are Clostridium. Facultative anaerobes are Kelbicella, symbiotic Rhizobium frankia, and Anabena azola. Associative symbiotic bacteria are Azospirillum, endophytic Glucanoacetobacter, phosphorus solubilizers are bacteria Bacillus megaterium and B. subtilis and B. circulans. Pseudomonas striata fungi are Penicillium species. Aspergillus and phosphate mobilizers are AM fungi, ectomycorrhizal fungi, ericoid mycorrhiza, orchid mycorrhiza, silicate and zinc solubilizers, plant growth promoting Rhizobacter, Pseudomonas species, and many more. Importance of fertilizers. Importance of biofertilizers are biofertilizers are known to make a number of positive contributions in agriculture. Supplement fertilizer supplies for meeting the nutrient needs of crop. Add 20 to 200 kg nitrogen per hectare by fixation under optimum conditions and solubilize and mobilize 30 to 50 kg of P2O5 per hectare. They liberate growth promoting substances and vitamins and help to maintain soil fertility. They suppress the incidence of pathogens and control disease, increase the crop yield by 10 to 15%. 50% and nitrogen fixers reduce depletion of soil nutrients and provide sustainability to the farming system. They are cheaper in price and pollution free and based on renewable energy resources. They improve soil physical properties, tilth and soil health. Some examples of biofertilizers are rhizobium, where rhizobia are soil bacteria living freely in soil and in the root region of the both leguminous and non-leguminous plants. However, they enter into symbiosis only with the leguminous plants by infesting their roots and forming nodules on them. Non-legume nodulated by rhizobia is a terma or parasponia species. The nodulated legumes contribute a good deal to amount of nitrogen fixed in the biosphere, 50 to 200 kg per nitrogen per hectare, varied with crops. Clover is 130 kg per hectare. Cowpea 62 to 128 kg nitrogen per hectare. Bejernik first isolated and cultivated microorganisms from the roots of legumes in 1888 and he named this as a Bacillus radicola and later modified as Rhizobium. Legume plants fix and utilize this nitrogen by working symbiotically with rhizobium in nodules on their roots. The host plants provide a home for bacteria and energy to fix atmospheric nitrogen, and in turn plant receives fixed nitrogen as a protein. Description and characteristics of rhizobium. Classification under family Rhizobiaceae, genus Rhizobium for stem nodulation, Rhizobium and Sanokibium. Morphology or unicellular cell size less than 2 micrometer, wide and short to medium rod, pleomorphic in nature, motile with peritracheous flagella, gram-negative bacteria, accumulate PHB granules. Nature, heterotropic in nature and symbiotic with legumes. Carbon sources are supplied by legume through photosynthesis, monosaccharides, disaccharides, and nitrogen sources are fixed atmospheric nitrogen. Contribution to the nature. Direct contribution of nitrogen symbiotically with legumes. Residual nitrogen benefits for the succeeding crops. Yield increases in by 10 to 35%. Improves soil structure. Produces exopolysaccharides. Produces plant growth hormones. Recommended for legumes, pulses, oilseeds, fodders. Promising strains NGR6, NC92, CC1 and CRR6, CRU14 and CO. Azotobacter. Another example of biofertilizer is Azotobacter. It is a free-living nitrogen fixer. The cells are not present on the rhizoplane but are abundant in the rhizosphere region. It is classified under the family Azotobacteriaceae. It requires more of organic matter and depend on the energy derived from the degradation of the plant residues. Bejernik was the first to isolate and describe Azotobacter. Description about this. Morphology: cell size large ovoid cells and size was 2 to 7 into 1 to 2 micrometers. Cell character is polymorphic in nature and gram character is negative. Physiology: nature chemoheterotropic, free living. Carbon sources are monodisaccharides, organic acids. Nitrogen sources: nitrogen through fixation, amino acids, ammonium and nitrate. Respiration is aerobic and growth is genus medium. Doubling time is 3 hours. Contribution to the soil: ability to fix atmospheric nitrogen 20 to 40 milligram BNF per gram of carbon source in laboratory, equivalent to 20 to 40 kg nitrogen per hectare. Production of growth promoting substances like vitamin B, indole, acetic acid, gibberellic acid. Ability to produce thiamine, riboflavin, pyridoxine, cyanoglobamine, nicotinic acid, pantothenic acid, etc. Biological control of plant disease by suppressing Aspergillus fusarium. Recommended for rice, wheat, millets, cereals, cotton, vegetables, sunflower and mustard and flowers. Another example for biofertilizers, Azospirillum. Azospirillum was isolated by Bejernik in Brazil from the roots of Paspalum in 1922 and named it as Azotobacter paspalii and later named as Spirillum lipoferum. Dubernier and Day reported the nitrogen fixing potential of some forage grasses due to activity of sulfur Spirillum lipoferum in their roots. Dubernier coined the term associative symbiosis to denote the occurrence of nitrogen-fixing bacteria in plants. Taxonomy was re-examined and Tarrant et al. Designated that these organisms as the Azospirillum. Contribution of Azospirillum to the soil. Benefits are promotes plant growth, increased mineral and water uptake, root development, vegetative growth and crop yield. Inoculation reduced the use of chemical fertilizer 20 to 50% and increased the efficiency to 20 to 40 kg nitrogen per hectare. Increases cost benefit ratio and reduces pathogen damage, inhibit germination of parasitic weeds, restoration of arid zone, margin mangrove ecosystem, reduces humic acid toxicity in compost. Recommended for rice, millets, maize, wheat, sorghum, sugarcane and for legumes. And another example, Gluconoacetobacter diazotropicus. It is an endophytic nitrogen fixer and formed to occur on large numbers in roots and stems, leaves of the sugarcane and other sugar-rich crops. It was first isolated from sugarcane. Cavalcanite and Dubernier reported this new endophytic nitrogen fixer and recently called as from G. diazotropicus. It can tolerate up to 30% of sucrose concentration and pH up to 3. Optimum sucrose concentration is 10 to 15%. Produce surface yellow pellicle on semi-solid medium. Does not grow at pH 7 and optimum pH is 5.5. Contribution of this bacteria to soil: fixes atmospheric nitrogen, production of Pg hormones, Ga and gibberellic acid and Ga in double that azoospermum, suitable for sugar-rich crops with acidic pH. And another example for biofertilizer is Azorhizobium. These genera can produce stem nodules. Stem nodulation has been reported in 3 genera of legumes: Escomina, Neptune, and Sesbanium. Stem nodulating rhizobium comprises both fast and slow growing types, having the generation time of 3 to 4 hours and 10 hours respectively. Those nodulate esquino can cross inoculate with S. rostrata strains, Azorhizobium colinodons. These fix nitrogen in free living conditions without differentiating into bacterioids. Have oxygen protection mechanism to fix nitrogen under free living condition. Mode of entry is through lateral root cracks. No infection threads is formed during colonization. Form both stem and root nodules in S. rostrata. Gram negative in nature and motile rods are formed. Produces root nodules in rice and wheat. And another example for biofertilizer we have taken is algal biofertilizer. Algal biofertilizers are the agronomic potential of cyanobacterial nitrogen fixation in rich rice fields. Was recognized in India in 1939 by D. Who attributed the natural fertility of tropical rice fields to nitrogen fixing blue-green algae. The rice field ecosystem provides an environment favorable for the growth of blue-green algae with respect to their requirements for light, water, high temperature and nutrient availability. Algal biofertilizers constitutes a perpetual source of nutrients and do not contaminate groundwater and deplete the resources. In addition to contributing 25 to 30 kg nitrogen per hectare, they biologically fixed nitrogen. They can also add organic matter to soil, excrete growth promoting substances, solubilizes insoluble phosphates, and amend the physical and chemical properties of the soil. Blue-green algae are a group of prokaryotic photosynthetic microscopic plants vigorously named as Myxopraceae, Cyanophaceae, Cyanobacteria. They show striking morphology and physiological similarities like bacteria and hence called as cyanobacteria. Contribution of this bacteria to the algal bacteria to the soil: important components of organic farming contribute 20 to 25 kg nitrogen per hectare, add organic matter to the soil, excrete growth promoting substances, solubilize insoluble phosphates, improves fertilizer use efficiency of crop plants, improve physical and chemical properties of the soil, reduces CN ratio, increases the rice yield by 25 to 30%. Cyanobacteria are more compatible with nitrate N than ammonia. And another example we have taken for biofertilizers are phosphate solubilizing bacteria. Phosphate solubilizing bacteria are beneficial bacteria capable of solubilizing inorganic phosphorus from insoluble compounds. Phosphorus solubilization ability of the rhizosphere microorganism is considered to be one of the most important traits associated with plant phosphate nutrition. It is generally accepted that the mechanism of mineral phosphate solubilization by PSB strains is associated with the release of low molecular weight organic acids through which their hydroxyl and carboxyl groups chelate the actions bound to phosphate, thereby converting it into soluble forms. Phosphate solubilizing bacteria have been introduced to the agriculture community as a phosphate biofertilizers. Phosphorus is one of the major essential macronutrients for plants and is applied to soil in the form of phosphate fertilizers. However, a large portion of soluble inorganic phosphates which is applied to soil as a chemical fertilizer is immobilized rapidly and becomes unavailable to the plants. Currently, the main purpose in managing soil phosphorus is to optimize crop production and minimize phosphorus loss from the soil. These are the types of phosphorus solubilizing bacteria that are Bacillus and Pseudomonas pigmentation we can see, and flagellated structures and colonies. Next, benefits of this all biofertilizers are: increase crop yield by 20 to 30%, generally replace chemical nitrogen and phosphorus by 25%, stimulate plant growth and activate the soil biologically, restore natural soil fertility, provide protection against drought and some soil borne diseases. Overall advantages of the biofertilizer use is cost effective and supplement to fertilizers, and they are eco-friendly, friendly with nature. These all fertilizer reduces the cost towards the fertilizers use, especially regarding nitrogen and phosphorus. Thank you.